We talk about anaerobic respiration. It's definitely not as efficient. If you recall, 39% of the energy that was available in glucose was passed on. In aerobic respiration, now only we get this 5%, so a lot less. Okay. And the biggest reason you can think about that is bonds have potential energy, and as fewer bonds are broken, less energy is going to be available for the reaction. Where does most of the energy end up? It ends up in these two products, which makes sense. If you compare ethanol here to the end products of aerobic respiration, which is carbon dioxide and water, ethanol we can use as an energy source. We don't really put carbon dioxide in our car as an energy source. So it's much less efficient, and we only net 2 ATP compared to 36 net. And that's the number we're going to use, remember, is 36 net ATP. We talked about how it could be up to 38, but the big thing is net gain of 36. All right, let's look at anaerobic respiration in more detail. Okay, what's the significance of all this? Now, glycolysis is a reaction that happens in all living things, okay? And we believe it evolved first, and you can think about why that would be. Number one, everything does it. And so it's likely that that came from a common ancestor a long time ago. Two, it's only a process that's truly anaerobic that can be done without the presence of oxygen. Therefore, that would have been more likely to evolve during a time when there was very little oxygen in the atmosphere. One of the things that we're going to learn in the third quarter is that on Earth's early atmosphere, oxygen was not present in a gaseous form in the atmosphere, which is significant. Number one, that needed to be that way. The reason we need no oxygen in the atmosphere was because oxygen is an oxidizer, and that as those first organisms would have evolved, that oxygen could have been extremely damaging to them. And so this idea is that glycolysis would have most likely came about when there wasn't any oxygen present, and that the later steps would have evolved when that would have been there. When we look at the process of cellular respiration, one of the things that we can think about is that, we use glucose. Glucose is a great model for us because it's easy to follow. We can see the exact number. If you take a class in college, they're going to take the glycolysis and cellular respiration, and they're going to use glucose as what enters into glycolysis. But just realize that glycolysis can accept any carbohydrates, not just glycolysis. So fructose could go into and get slightly different results, and get different reactions. Okay. Galactose could as well. In addition, proteins could also enter into this process in the form of amino acids. Okay, so the amino groups could be fed into glycolysis or the citric acid cycle as well. So we can actually get energy, ATP, directly from protein breakdown as well. Fats could enter into this if we break them down in those glycerol groups. So if you remember, a fat was a triglyceride and a fatty acid tail. The glyceride and the fatty acids could be used. The fatty acids would be used to make acetyl-CoA. Glycerol would be used in glycolysis. So the idea is this, that when we look at cellular respiration, we look at this process, we only use glucose. That's our baseline. What would be important to know for the test? Just realize I'm not going to ask you these specific things. These are examples. What do you need to know? Know that other things can enter. Proteins could enter. Fats can enter. Carbohydrates can all enter into this process. So we're looking at it just as a glucose, as a model, as an introductory unit, but do realize that cellular respiration can use these other products as a source of the energy. And the energy is going to come from those potential energy in the form of those bonds. I like this diagram. It's not just because there's pizza on there. It happens to be pepperoni and sausage, my favorite. But when you take a look at this, basically what it shows is the model that we used in class was we used glucose. And so basically this is the pathway that we used, right? I mean, if you look at it, that's exactly what we did. We took glucose into this process. We took glucose, sent it into glycolysis. Glycolysis converted to pyruvic acid and pyruvate. It was then converted into acetyl-CoA, giving off some NADH that can go into here. Some NADH was produced there. That gets fed into the citric acid cycle, producing ATP, more NADH and FADH. All of that was combined to make ATP. okay? So the idea is this. Well, carbohydrates can do all that, and that's fine, but there's other things as well. And so when we take a look at this process, there's other things that can enter in. If you take a look at fats and oils can actually have a little bit higher energy a pound because there's more molecules in there, a pound of fat and oil, and it can enter in to this process as well, generating ATP as well, depending on how it enters in. Fatty acid goes into glycerol, and once you get into the system, you can continue down. So the fatty acid tail will go right to this stage, go right in the citric acid cycle, and be able to go down that way. And so the basic point is we can get a lot of energy from just more than our carbohydrates. Proteins, when you break them down, are broken down in amino acids. And when we do that, most of the time, we take those amino acids and make new proteins out of them. So I eat some beef. That protein that's in the meat is actually broken down in amino acids, and then my body will use those amino acids to make other proteins my body needs, maybe some enzymes, maybe some structural proteins for the cells. When there's a lot of amino acids available, we can actually use those amino acids as an energy source. And so if you take a look here, that amine structure there, that's the NH2, can actually be a product or energy source. Once again, what's important for the test? Know that amino acids can be a source of energy as well. This, to me, would be a great multiple choice question. That's a mark all would apply. People always say, oh, well, these are so challenging. Well, take a look at it. What am I basically telling you? I'm telling you that, yes, we only looked at carbohydrates as a source. We only looked at glucose as a source, but other things, including proteins, can be the source of cellular respiration. So just think about that. If you're going to use proteins for cellular respiration, now this is a complicated diagram. Most people look at that and go, crap, I don't know most of this stuff. Just realize what these are telling you. These are different types of amino acids on here that'll kind of enter in at different spots. But basically the idea is this, is we got to remove an NH3, okay? And then that NH3 is converted into urea and excreted in urine. And so when we do this process, a lot of times when we develop proteins, that's where urea comes from. And one thing that you'll notice is if you do eat a lot of meats, you tend to produce a higher amount of urea, a higher amount of ammonia, which eventually gets converted into urea in your urine, and it can even cause like a slight change in the smell of your urine for that reason. If you eat tons of proteins, or if you go home tonight and eat a bunch of steak and then more meats, not a lot of carbs, your body's going to take those extra amino acids and get energy out of them, and the process release that NH3, which will be converted to urea. Once you remove that, the amino acids can enter into it at different points. Basically, again, just realize that we can use proteins as a source of energy. All right. This last two slides here real quick are just about how do I control this process. So when you think about it, the regulation of cell respiration, how do I control it or prevent it maybe from occurring? What if I don't want to break down amino acids? What if I don't want to break down every sugar that I get? It uses a feedback mechanism. Now think about that word, feedback. If you're talking to me, right, and I give you feedback in class, basically I'm kind of observing, but I'm going to say something to you that the goal is to hopefully change your what you're doing or what you're thinking or how you're acting. So the idea is this. If ATP starts to drop, what it will do is we'll actually speed up respiration. And when there is a lot of ATP, respiration slows down. So in other words, ATP acts as a feedback, okay? So the more ATP we have that's available, we'll start slowing down. We don't need to kick this into high gear. And so basically, how do we do that? Well, by the amount of ATP will affect different enzymes. And the different enzymes, throughout this whole process, we talked about how important enzymes were. We didn't say specifically the names of any specific enzymes, but we just said there's a lot of enzymes running this reaction. And the idea is if we have some ATP present, it will actually affect and cause those enzymes to not work as well. Their feedback affects the ability of the enzyme to do its job, therefore slowing down the enzyme. I'll show you here on the next slide. All right, so this slide here is a good example. What ends up happening is this. If you take a look here, this enzyme, fructokinase, is important in glycolysis. It kind of gets everything going. The idea is if we have AMP, that's going to really stimulate. Well, why would AMP? Well, if we have a lot of AMP available in the cell, that means that our ATP has been used and it's been broken down to ADP. And we're still needing energy, so some of that ADP has been broken down just because it's so desperate, producing AMP. That AMP tells us that we're really short on ATP, so it stimulates or increases the ability of this enzyme to do its job, breaking down the glucose to run this reaction. Okay? Now, what causes this phosphofructokinase to slow down? ATP will actually inhibit this. So a lot of ATP will cause this to slow down, and therefore slowing down the entire process. Okay? There's another compound called citrate, which is produced in the citric acid cycle, which can also slow this process down as well. So the idea is this. The only enzyme that you need to know for our test at all, completely, I won't ask you any other enzymes, and I won't do it in a fill in the blank, it'll be a multiple choice, is right there. Just realize that is the enzyme that is regulated or controls this reaction. AMP causes the reaction to speed up. Too much citrate or too much ATP in the cell will cause this enzyme to slow down. That's it. Don't make it more complicated than it needs to be. Just know that this enzyme is what runs the ship. If we're so short on ATP that we had to create ADP and then eventually AMP, boom, that's going to stimulate a lot of this to start kicking in and working, getting the process to work. If the cell's a lot of ATP available, we should continue this process, slow it down a little bit. So because there's a lot of ATP available, it'll actually cause this enzyme not to work as efficiently. Citrate is produced here when the oxaloacetate combines with acetyl-CoA to create citrate. If we have a lot of citrate available, that means that this process is running really efficiently and producing too much product. It'll come back in here and slow that down as well. So it's a way to inhibit the process. If we want to slow this down for some reason, we don't want to burn up all our glucose at once, okay? It seems complicated. It's really not. Just realize there's an enzyme that allows us to speed up production or slow down the production of ATP, and it's controlled in a feedback mechanism. The ATP and citrate slow it down, AMP speeds it up. All right, that's all I got for today. Hopefully you're ready for the test. As I said, plan on a mark all apply question that asks what type of molecules can go into, that can be used in cellular respiration. It'll be something like glucose, amino acids, glycerols and fatty acids, and all of those things can. So it'd be a mark all apply, it'd be all of them. Also expect a question on this slide here, something about what enzyme is responsible for feedback inhibition or regulation of this process. That's your answer right there. And then I might ask you what would speed up that process. AMP, presence of AMP will speed it up. And what will slow down that enzyme? Too much ATP and too much citrate. Okay? There's a couple freebies for you for watching the video. Thanks and have a good day.